friends, I am Romila Lemos, Assistant Professor, Department of Forensic Biology, Institute of Forensic Science, Mumbai. Today's topic of our discussion and learning is all about Forensic Serology, Part 5. We will be discussing the following modules today. Module 1, Introduction to Blood. Module 2, Properties of Human Blood. Module 3, Collection, Preservation and Dispatch of Blood Sample. Module 4, Preliminary Examination of Blood. Module 5, Preliminary Examination of Blood. Continued. Module 6, Factors Affecting Preliminary Examination of Blood. Module 7, Conclusion. Since blood evidence associated with a crime can provide information that may solve the case, it is essential to correctly document, collect and preserve this type of evidence. Improperly handled blood evidence can weaken or destroy a potential source of facts in a case. Properly collected and preserved blood evidence can establish a strong link between an individual and a criminal act. Blood evidence or the lack of blood evidence can also be used to bolster or contradict a witness statement or any statements that the suspect may make. Blood evidence can also point the investigator in the direction he or she needs to go to solve the case. If blood evidence is documented, collected and stored suitably, it can be presented to a judge or jury several years from the time of the criminal act. The most powerful application of blood evidence is the ability to absolutely eliminate a person as a potential suspect in a crime. The human blood constitutes about 8% of the body weight of a healthy individual. The fluid portion of the blood is known as the plasma, while the cellular portion of the blood consists of red blood cells, white blood cells and platelets, all of which are suspended in the plasma. The red blood cells. These cells are also called as erythrocytes. Their lifespan in humans is approximately 3 to 4 months. Additionally, mature human erythrocytes do not have a nucleus and therefore lack the nuclear DNA. Erythrocytes consist of hemoglobin, proteins that are responsible for the transportation of oxygen. Most adult human hemoglobin consists of 4 polypeptide chains, 2 alpha chains and 2 beta chains. Thus, adult hemoglobin is designated as alpha 2, beta 2. Other forms of hemoglobin are also present. Under normal physiological conditions, each hemoglobin subunit contains a heme moiety that binds to oxygen. A heme molecule consists of an organic component known as protoporphyrin 9 and a ferrous Fe2+ ion. A heme molecule is also known as ferroprotoporphyrin. The ferrous ion of heme forms 4 bonds with the nitrogens of protoporphyrin 9 along with a 5th bond with the hemoglobin chain and a 6th bond with a molecule of oxygen. Other chemicals such as carbon monoxide and cyanide may also bind to the ferrous ion of the heme molecule and can cause chemical asphyxia. Heme groups are also present in the blood of various animals and in other proteins such as myoglobin in muscles and neuroglobin in the brain. White blood cells. These are also called as leukocytes and are subdivided into 3 types, that is the granulocytes, the lymphocytes and the monocytes. White blood cells are involved in defending the body against infection. They have a nucleus and thus represent the main sources of nuclear DNA from the blood. The third type of cells are the platelets. These are also known as the thrombocytes and they play a role in blood clotting. Platelets aggregate at sites of vascular and blood vessel injury. Like erythrocytes, they too lack nuclei. Once the crime scene has been thoroughly documented and the locations of the evidence noted, then the collection process can begin. The collection process usually starts with the most fragile or most easily lost evidence. Special consideration needs to be given to any evidence or objects that need to be moved. Collection then continues along the lines of the crime scene, trail, or in some other logical manner. Photographs should be continually taken if the investigator is revealing layers of evidence that were not previously documented because they were obscured. Most items of evidence are collected in clean, unused paper containers such as packets, envelopes, and bags. Moist or wet biological evidence from a crime scene are collected in clean, unused plastic containers at the scene and transported back to an evidence receiving area if the storage time in sealed plastic is less than 2 hours. This is done to prevent contamination of the other evidence. Once in a secure location, wet evidence, whether packaged in plastic or in paper, is removed and allowed to completely air dry. That evidence can then be repackaged in a new, clean, unused, dry paper container. Care is to be taken so that no evidence containing moisture is sealed with plastic or paper containers for more than 2 hours. Moisture allows the growth of microorganisms that can destroy or alter the evidence. Blood evidence must never be exposed to excessive heat or humidity. If possible, the blood-stained evidence is refrigerated until it can be transported to the crime lab. The evidence should also be taken to the lab as soon as possible. The collection and preservation of blood-stained evidence is important because this evidence can be used to type blood or run DNA analysis. There are two different types of blood that can be collected at a crime scene, that is the liquid blood and the dried blood. Liquid blood evidence is generally collected from blood pools but can be collected off of clothing as well, using a gauze pad or a sterile cotton cloth. Once the sample is collected, it must be refrigerated or frozen and brought to the laboratory as quickly as possible. The sample must first be dried thoroughly at room temperature. It is important to get the sample to the laboratory as quickly as possible because after 48 hours, the sample might be useless. If the sample has to be mailed, it should be completely air dried before packaging. If the sample isn't completely dry when it needs to be packaged, the sample should be rolled in paper and labeled and then put into either a brown bag or a box. The paper bag or box is then sealed and labeled again. It is important to only place one item per container to avoid contamination, and samples should not be placed in plastic containers. Samples shouldn't be in plastic containers because if the sample is still damp, the moisture from the sample can cause the microorganisms to grow that can destroy the evidence. Also, due to this fact, samples should not be in any container for more than 2 hours. Also, due to this fact, samples should not be in any container for more than 2 hours. Dried blood stains can be found on small objects, larger objects, and on clothing. When dried blood is found on a small object, the entire object can be sent to the lab after it's been properly packaged and labeled. When dried blood is found on a larger object that is transportable, an investigator should cover the stained area with paper and tape the paper to the object to avoid contamination. If the object that is stained is not transportable, there are different ways that an investigator can collect the sample. One option is to cut out the stained area of the large object. If the portion is cut out, the sample is packaged the same way as described above, but a control sample should also be provided in a separate package. Another option is to use fingerprint tape and lift the sample as well as the surrounding control area. If this method is used, it is important for the investigators to not touch the sticky side of the tape with bare hands, and the investigator should run an eraser or some type of blunt object over the placed tape to ensure that contact is made with the dried stain. The lifted stain is then to be packaged and labeled, then delivered to the laboratory. Another way to collect a sample of an object is to use a clean, sharp object to scrape flakes of the stain into a paper packet. The last two methods of collection of a dried blood stain on a large object require the use of distilled water to dampen the stain before rolling a thread in the stain or absorbing the stain with a cotton square. These two methods are not recommended due to the risk of contamination. When dried blood is found on clothing, the entire article of clothing should be packaged and labeled and delivered to the lab. It is important for the investigator to remember to keep each sample separated so that there is no contamination between the samples. Let us look at the mechanisms of presumptive assays. Presumptive blood assays are designed to detect traces of blood. These assays are based on the basic principle of the oxidation-reduction reaction catalyzed by the heme moiety of the hemoglobin. As a result, colorless substrates catalyzed by heme undergo an oxidation reaction causing either chemiluminescence, fluorescence, or a change of color. These assays are very sensitive and can detect blood in the samples with 10⁻⁵ to 10⁻⁶ fold dilutions. A positive reaction indicates the possible presence of blood. Additionally, most of these assays do not interfere with forensic DNA analysis. The following are the presumptive tests for blood. A presumptive test, when positive, allows a qualified conclusion that blood is present in a tested sample. When negative, the test often helps to eliminate stains that need not receive further consideration. In the event of a positive test and when sample quantity is sufficient, further action is usually taken to confirm the presence of blood because no single test is absolutely specific for blood. Presumptive tests may be recognized as those that produce a visible color reaction or result in the release of light. Both types rely on the catalytic properties of blood to drive the reaction. The oxidation-reduction reaction between blood and the substrates involves a change in the oxidation state of a molecule. Specifically, the oxidation of a molecule means that the molecule has lost electrons, and the reduction of a molecule means that the molecule has gained electrons. Chemicals that can be reduced and therefore gain electrons from other molecules are called as oxidants, and in contrast, reductants are chemicals that can be oxidized and therefore lose electrons to other molecules. In biochemical reactions, oxidation often coincides with the loss of hydrogen. In presumptive assays, heme is utilized as the catalyst and the hydrogen peroxide is utilized as the oxidant for the reaction. In the presence of heme, a colorless substrate is oxidized, yielding a product with color, chemiluminescence or fluorescence. Let us look at the colorimetric assays. Many procedures are available for detecting heme in blood through color reactions. The most common agents are phenolphthalein, leucomalachite green and benzidine derivatives. The color reactions produced by these assays can be observed immediately with the naked eye. Phenolphthalein assay. Phenolphthalein, a member of a class of indicators and dyes, is used in titrations of mineral and organic acids as well as most alkalis. The phenolphthalein assay for blood identification is also known as the Kastle-Meyer assay. In this reaction, phenolphthalein, a colorless compound, is catalyzed by heme with hydrogen peroxide as the oxidant. The oxidized derivative is phenolphthalein, which appears pink under alkaline conditions. The second assay is the leucomalachite green assay or the LMG assay. Malachite green is a triphenylmethane dye. The leuco-base form of malachite green is colorless and can be oxidized by the catalysis of heme to produce a green color. The reaction is carried out under acidic conditions with hydrogen peroxide as the oxidant. The third chemicals that can be used are benzidine and derivatives. Historically, benzidine was used as an intermediate in dye manufacturing. Subsequently, it was used as a presumptive assay for the presence of blood after the discovery that the oxidation of benzidine can be catalyzed by heme to produce a blue to dark blue color carried out in an acid solution. Since the blue color may eventually turn brown, the reaction must be red immediately. Benzidine was however found to be a carcinogen and is therefore no longer used for forensic testing. Orthotolidine is a dimethyl derivative of benzidine. Its oxidation reaction can be catalyzed by heme to produce a blue color under acidic conditions. Orthotolidine is also considered a potential carcinogenic based on the animal studies, and for this reason it has been replaced by tetramethylbenzidine. Tetramethylbenzidine or TMB is a tetramethyl derivative of benzidine. The oxidation of TMB can be catalyzed by heme to produce a green to blue-green color under acidic conditions. TMB continues to be used. The hemastix assay is a TMB based assay that utilizes a TMB containing strip device. A test is carried out by applying a moistened sample to a hemastix strip. The appearance of a green or a blue-green color indicates the presence of blood. Often, the presence of blood is suspected based on witnessed information or expected in a particular location, but under normal lighting and viewing, little can be seen. A drag pattern across a floor that was cleaned or washed away, or a washed spatter pattern on a wall might be typical examples. At this point, luminol and fluorescent tests come into play. These are organic compounds whose oxidation products have chemiluminescent or fluorescent properties and are subsequently utilized for testing. In the chemiluminescence assay, light is emitted as a product of a chemical reaction. In this category, luminol produces chemiluminescence when blood is present. In contrast, a fluorescence assay requires the exposure of an oxidized product such as fluorescent to a particular wavelength of excitation light source. The fluorescence is then emitted at longer wavelengths than that of the excitation light source. One advantage of chemiluminescent and fluorescent reagents is that they can be sprayed over large areas where latent blood stains are potentially located. A positive reaction identifies blood and also reveals the patterns of bloody impressions such as footprints and fingerprints. These methods are very sensitive and can pinpoint the locations of even small traces of blood. They are useful for detecting blood at crime scenes that have been cleaned and show no visible staining. The presence of blood and the patterns displayed by blood can provide information of value, and one should be alert to both. Further, the nature of these tests makes them potential sources of contamination of the blood. Therefore, if a stain can be seen and collected, these tests probably should not be used. Since they add material to the tested area, their use should be carefully considered, often as a last resort. They are of more value in locating and defining areas of possible blood than in specifically identifying it. One disadvantage of chemiluminescent and fluorescent reagents is that precautions must be taken if stains are very small or have been washed. The spraying of the presumptive assay reagents may further dilute a sample and thus lead to difficulty in isolating sufficient amounts of DNA for forensic DNA analysis. Both luminol and fluorescent are classified as irritants. Although neither compound is classified as a carcinogen, safety recommendations are prescribed for both and they should not be treated carelessly. Let us look at the luminol reaction. Luminol or 3-aminophthalhydrazide is usually utilized as a chemiluminescent reagent. The oxidation reaction of luminol catalyzed by heme produces light in the presence of an oxidant. The catalytic activity of the heme group accelerates the oxidation of the luminol, producing a blue-white to yellowish-green light depending on the reagent preparation if the blood is present. The light emitted from a positive reaction can only be observed in the dark, which limits the applications of luminol. The forensic application of luminol at crime scenes involves spraying a mixture of luminol and a suitable oxidant in an aqueous solution over the area thought to contain traces of blood. A resultant blue-white to yellowish-green glow will indicate a blood presence. Outlines and details are often visible up to 30 seconds before additional spraying is required. The photo documentation of a luminol enhanced pattern should be done immediately before it fades away. Excessive spraying, however, usually results in the diffusion of stain patterns. The greatest advantage of luminol is the sensitivity. It has been found that luminol can detect hematine from hemoglobin in dilutions up to 1 in 10 million. The next molecule is fluorescent. Fluorescent is another reagent that is used to test for the presence of blood stains at a crime scene. When oxidized and catalyzed by heme, fluorescent demonstrates fluorescent properties. The catalytic activity of the heme accelerates oxidation by hydrogen peroxide of the fluorescent to fluorescein, which then fluoresces when treated with UV light. Usually, fluorescent sprayed stains are exposed to light in the range of 425 to 485 nanometer using an alternate light source device. In a positive reaction, the oxidized fluorescent emits an intense yellowish-green fluorescent light which indicates the presence of a blood stain. The light emitted from fluorescent sprayed stains lasts longer than that of luminol. As with luminol, careful considerations of the ultimate aim of the investigation should guide fluorescent application at a scene. If the blood at the scene can be observed and collected without the aid of fluorescent, then use of fluorescent may be unnecessary. On the other hand, if the purpose is to define and enhanced patterns not visible but thought to be present, then any visible blood, desired or subsequent analysis can be covered to shield it from chemical contamination or secured before the area is tested. One major advantage of fluorescein is that it does not fluoresce with household bleach, whereas luminol does react. The catalytic assays discussed in the previous sections are not specific to blood alone, which can possibly lead to the observation of false positive or false negative results. A few of the factors affecting the results are oxidants. Chemicals that are strong oxidants may cause a false positive reaction. Such chemicals can catalyze the oxidation reaction even in the absence of heme and result in a false positive reaction. Certain metal salts such as copper and nickel salts, household bleaches and cleaners that contain hypochlorite ions, and hair coloring products that contain hydrogen peroxide work as oxidants. To address this problem, a two-step catalytic assay should be performed. The substrate is applied first to the sample in question. A color change occurring before the addition of hydrogen peroxide indicates a false positive result due to a possible oxidant in the sample. If a color change is observed after the addition of hydrogen peroxide, the result is a true positive. The next substances which can affect the results are plant peroxidases. Many types of plants such as horseradish contain peroxidases. Plant peroxidases may also catalyze oxidation reactions and lead to false positive results. However, plant peroxidases are usually heat sensitive and may be inactivated by high temperatures. Because the heme molecule is relatively stable at higher temperatures, samples can be retested after heating. This will inactivate any plant peroxidases in a sample. The other interfering agents include reductants. Although not common, a false negative result can occur when a strong reductant is present in a sample. Strong reductants such as certain metal ions, including lithium and zinc, may inhibit the oxidation reaction. Thus, the collection and identification of blood evidence, preferably at the crime scene, plays a vital role in determining its evidentiary value. While most of the preliminary assays are simple, rapid and sensitive, they still do not confirm the presence of blood owing to a number of interfering factors. Therefore, confirmation of blood evidence requires another round of assays which show high specificity towards blood. The appropriate approach for identification of blood thus employs a presumptive chemical screening test that is frequently followed by a confirmatory test to clearly establish the identification. A visual observation of an untested stain coupled with positive chemical presumptive and confirmatory tests will provide sound data to support the identification. A visual observation of an untested stain coupled with positive chemical presumptive and confirmatory test will provide sound data to support the identification. Thus, we come to the end of our next chapter, that is Forensic Serology Part 5. You are now left to do self-study in the above mentioned topics. Thank you. Goodbye.